Hello students, welcome to CBSE NCERT Solutions by Vedantu. I am Ms. Kopika, your own biology master teacher. So today we are going to do the chapter Excretory Product and its Elimination. Is one of the very important chapters of your human physiology. So I hope all of you have loved the series of NCERT line by line, and you can definitely let us know in the comments how you're finding these series where we go every line of NCERT and help you understand every single line of NCERT as well. So yes, we are going to do the next chapter, that is Excretory Product and Elimination. We are going to do some parts of it. So before that, let me also check the volume and if we are live yet. Okay, just give me a minute. Also check the volume and if we are okay, perfectly audible and visible. Hello Sony. So students, we will do excretory product and elimination line by line. So let's see what's the first paragraph telling. Animals accumulate ammonia. Okay, animals accumulate ammonia, urea, uric acid, carbon dioxide, water, ions like sodium, potassium, chlorine, phosphate, sulfate, etc., neither by a metabolic activity or by other means like excess indigestion. These substances have to be removed totally or partially. In this chapter, you will learn the mechanism of elimination of these substances with special emphasis on common nitrogenous waste. Ammonia, urea, uric acid are major forms of nitrogenous waste excreted by the animals. Ammonia is the most toxic form and requires large amount of water for its elimination. Okay, most toxic form and requires large amount of water for its elimination. Yes. Happy Gandhi Jayanti to all of you. Whereas uric acid, being the least toxic, can be removed with a minimum loss of water. A minimum loss of water. Okay, so students, here what they're telling in the first paragraph is our body makes waste. Okay, be it a smaller organism, be it a larger organism, there is some amount of waste that is formed in our body. Now that waste has to be removed out. Now for the removal of the waste, there is a process that is called your excretion. Now what happens is, when students are studying excretion, since digestion is deleted for the NEET students, or generally let's say students misunderstand that excretory product, okay, or excretory system is going to remove the undigested food. Okay, excretory system is going to remove the undigested food, which is absolutely not true, because your digestive system, okay, your digestive system is removing the fecal matter. Fecal matter is being removed by your digestive system. And excretory system, on the other hand, hi Tanuja, hi Divya, okay, on the other hand, excretory system is removing out your urine. So this is the difference. One system is there to make the urine, another system is there to remove the fecal matter. So this misunderstanding students usually have while learning about excretion, because at a very smaller age we always study excretion means removal of something. Okay, now so removal of nitrogenous waste. Now that nitrogenous waste can be ammonia, urea, uric acid and other substances. Now here you're going to learn that depending on what you're excreting out, how much toxic is it, how much water does it require, and how is the mechanism. Okay, fine. Yes. Okay, perfect. Now students, here they're mentioning that ammonia is the most toxic. Okay, ammonia is the most toxic and it requires a lot of water to be removed out. So you just imagine that if ammonia is the most toxic and it requires a lot of water to be removed out, which kind of organisms can opt for ammonia? Like opt means it's not like before you were born you have a selection process where you can select which you want to excrete out. Not at all. I'm just asking, like, generally, which organism do you think will capable of giving out ammonia? It will be organisms who are underwater. So if I need a lot of water to excrete out something, okay, that means I should be an aquatic organism mainly. Now the process of excreting ammonia is called as ammonotelism. What is it called? Ammonotelism. Many bony fishes, aquatic amphibians and aquatic insects are ammonotelic in nature. Ammonia, as it is readily soluble, is generally excreted by diffusion across body surface or through gill surface. Okay, so ammonia, how students? The process of excreting ammonia is called as ammonotelism. Okay, so the organism that excrete out ammonia is called as ammonotelic. So telism is the process, telic is the organisms. Okay, ammonia is readily soluble and is generally excreted out by diffusion across body surface or through gill surface, especially in fishes, as ammonia ions, okay, ammonium ions. Kidney do not play a significant role in its removal. Why doesn't kidney play an role? It's very simple, students. Imagine something is very toxic, okay, something is very toxic to in our body, and we do not have enough time. See, when kidney is doing something, it's going to take its own time and make the urine and etc. like that. Now, since this is so toxic, ammonia is so toxic, we do not have enough time to give kidney to play the role. So what we are going to do, we will not do anything. You be removed out from the body surface. Understood? Now, terrestrial adaptation necessitates the production of lesser toxic nitrogenous waste like urea and uric acid for conservation of water. And not only that, terrestrial organisms, the excreta is going to be mixed with the soil. So we cannot excrete out very toxic substances like ammonia on a daily basis in the soil, because this is going to dissolve in the soil and we are going to use it. At the end, it's going to be a cycle that is going to come to us. Correct? So we cannot use that as well. Okay, we cannot use that as well. Now, mammals, many terrestrial amphibians and marine fishes mainly excrete urea and are called as uricotelic. So now we studied ammonia, ammonotelic. Next is urea, uricotelic. Ammonia produced by metabolism is converted into urea. So sometimes in our body or any ureotelic body, there could be ammonia that is formed. Now that ammonia will be immediately converted into urea. Okay, ammonia will be converted to urea with the help of Ornithin cycle. What cycle? Ornithin cycle. Got it? Ornithin cycle. So this Ornithin cycle, or else simply you can call it as the urea cycle as well. Urea cycle, this happens in our body, okay, in the liver it happens, and in the liver this cycle is going to help in converting ammonia to urea immediately. Okay, converting ammonia to urea immediately. Now students, why this conversion is done? For the simple reason, because ammonia again is very toxic. Yes or no? Ammonia is very toxic and urea is less toxic. So if a very toxic substance is in our body, then definitely our job is to convert this toxic substance and make it less toxic. Okay, so uricotelic, ammonia produced by metabolism is converted into urea in the liver of these animals and released into blood, which is filtered and excreted out of the kidney. Some amount of urea may be retained in the kidney matrix of some of these animals to maintain a desired osmolarity. Now this you will study in the last concept of this chapter, which is called as vasa recta. Okay students, what I'm writing is going out. 1 second. Wait, let me just check now. Someone has changed my settings in the laptop. 1 second, let me just check how it is coming now. I think it's the same, no? Okay, just give me 1 minute. I'll just fix this. Okay. Okay, I have to write a little inside. Okay, got it. Okay, got it. Fine. So students, here, so some amount of urea is going to stay in our body. Okay, some amount of urea is still going to stay in our body. Why? Because of the fact that urea is going to help in osmolarity. So I think most of you already know this, that you will learn this when you are learning about the vasa recta, that is your counter current mechanism. Okay, counter current mechanism. When you learn about counter current mechanism, you are actually going to learn this. This is inside only, no? Yeah, counter current mechanism. So some amount of urea may be retained in the kidney matrix of some of these animals to maintain desired osmolarity. Now reptiles, birds, large snails, insects excrete nitrogenous waste as uric acid in the form of pellet or paste with a minimum loss of water and are called uricotelic organisms. So students, all of you remember when we go out, exactly when we are wearing white, okay, here there is a a pigeon who is attracted to white. Okay, in my area there's a pigeon who loves white. So what this pigeon does is it always, okay, it always you know, puts his pillet, okay, on the white cloths, okay, on the white cloth. So that is a very sad thing. Yeah, which is a very sad thing, because we cannot wash any more white clothes, because we have to wash it and hide it and keep it. Okay, so there are some animals which like some colors. So students, what did we learn so far? We learned that ammonia, okay, ammonia is an excretory substance, and the process of ammonia coming out is called as ammonotelism. How did you guys spend your day today? Did you all revise something? Ammonotelic, okay, organism. Monotelic organisms. So if you have some experience like this with the bird, you should let me know. Okay, now students, ammonia is readily soluble. Okay, readily soluble. Now not only readily soluble, they require a lot of water. Okay, lot of water for removal. Morning physics, afternoon biology, evening bio. Oh, lot of water for removal. Bio is a full-time person, no? Yes, lot of water they require for removal, and not only that, they kidney, okay, kidneys role. Kidneys, we can tell lack of role of kidney. That means kidney is not going to come for help. Okay, then students, what else? This requires a lot of water. The examples are going to be, okay, the examples are going to be all the aquatic organisms, including aquatic arthropods. Okay, aquatic organisms, aquatic arthropods, all this, aquatic arthropods, etc. Fishes, okay, fishes, all of these are going to excrete out urea. Okay, excrete out urea, I'm sorry, ammonia. Now next one is urea. Urea is excreted mainly by us mammals. Now the thing with urea is less toxic than ammonia. Okay, less toxic than ammonia. So the red flag in this group is ammonia. Okay, she is the red flag, he is the red flag, ammonia is the red flag. Okay, ammonia is the red flag. Why ammonia is the red flag? Because this is the most toxic. This is your Gen Z language, no? Red flag, blue flag, green flag, you know. He is a green flag. So you people tell this, no, to each other. So less toxic than ammonia. This is also soluble in water, and it requires less water. Okay, it requires less water. Total role of kidney. Okay, kidney's function is there. The kidney has a role. Okay, next thing is who are the organisms who are going to have it? Is your mammals, okay, amphibians, adult amphibians. So in larval stage, amphibians are going to give out ammonia only. Okay, adult amphibians. So here you can write larva of amphibians or larvae of amphibians. Okay, and here mammals, adult amphibians, all of this are going to come back. Okay, here. Then you have uricotelic, and now these organisms are called as, wait, these organisms are called as ureotelic. Okay, the spelling, many students confuse with uric acid and urea. Okay, process is this. Then uric acid is going to be uricotelism. Got it? Co is going to come. Uricotelism and uricotelic organisms, and these are going to come out in the form of semi-fluid, okay, pellet or semi-fluid paste. All of you at least once would have got some bird's blessing, so I don't have to explain much. And students, uric acid, you can ask me a question, ma'am, uric acid is the least toxic among these people. Uric acid is the least toxic. Why are we not using it, or why are we not excreting out uric acid when it's the least toxic? That is because, students, uric acid requires a lot of ATP. Okay, it's an expensive process. It's an expensive process and we cannot afford it. Expensive process means not like price is too high here and there, not like that. Price here, the main issue is that we cannot afford this one because we do not have that much energy to excrete out. Clear? Okay, fine. It all filled up in the same sheet only. Okay, done. Chalo, very nice. Okay. Yes. So this is your urea. Got it? Now we will continue. Next one. A survey of animal kingdom presents a variety of excretory structures. In most of the invertebrates, these structures are simple tubular forms, whereas vertebrates have complex tubular organs called as kidneys. Some of these structures are mentioned here, like protonephridia or flame cells. All my students should be able to answer this. Protonephridia or flame cells are the excretory structures in platyhelminthes, right? We recently only studied animal kingdom. Flatworms. Flatworms like your planaria, taenia, solium, okay, your liver fluke, all of these organisms is going to have protonephridia or flame cells, okay? So in your NCERT, flame cells is mentioned when we learned animal kingdom. So this is the first one, okay? Protonephridia or flame cells are present. Got it? 1 second, students. Oh, mix up हो गया. Okay. Now, rotifers, some annelids and cephalochordates like amphioxus. Protonephridia primarily concerned with tonic and fluid volume regulation and osmoregulation. Nephridia are a tubular excretory structure of earthworm and other annelids. Okay. Some annelids are exceptions, but some annelids have nephridia, some annelids have protonephridia. Okay, understood? Now, nephridia helps to remove the nitrogenous waste and maintain a fluid and ionic balance. Malpighian tubules are excretory structures of most of the insects, including cockroaches. What is the structure? Malpighian tubules. Malpighian tubules are excretory structure in most of the cockroach. Okay, arthropods. Malpighian tubules help in removal of waste and osmoregulation. Antennal glands or green glands perform the excretory function. So now this box you should learn. After that, you do not have to worry at all. Okay, I think most of you would know now by now. Okay, let's start. So let's write the organism, okay, or the excretory organ. Excretory organ. Just make a box and keep it in your NCERT. So whenever you're learning, you'll always look into the box instead of searching for which page, going to animal kingdom and searching. So don't do that. Always make a note of what is very important to you. Okay, this is a very beautiful line I could draw possibly. So yes, excretory organ. So we'll start with what is given first to us. Protonephridia. Okay, protonephridia or flame cells. Protonephridia or flame cells. Where organ is going to be, what is the organ, students? Platyhelminthes. Okay, platyhelminthes, or you can write flatworm instead of this. Okay, flatworm. Some of the rotifers and annelids. Some rotifers and annelids. These are all going to have your protonephridia or the flame cells. Next is going to be, okay, next is going to be nephridia. Okay, next is going to be nephridia. Nephridia, the answer, okay, here, wait 1 second. Yes. Nephridia is going to be earthworm. Okay, earthworm. Next one is going to be Malpighian tubules. Malpighian tubules. It is going to be your arthropoda. Some arthropoda, okay, especially your cockroach is mentioned. Cockroach. Okay, last one, last but not the least, green glands or anterior glands. Names are there, okay, for the anterior glands, green glands. Structure is going to be prawns. Some crustaceans you can write, okay, some crustaceans you can write, but mentioned here is prawn, but I'm writing in bracket crustaceans. Like a lobster, all of this can come under this. Okay, all of this can come under this. So students, this box is very, very important. I hope the box is clear also. Yes, very nice. Okay, it's very clear. So excretory organs, this is from your animal kingdom itself, but it's very, very important. Okay, very, very important. They can give you easily MCQs from this question. Chalo, done. Now we're going to start human excretory system. So before that, we have some things to learn about the kidney, the size of the kidney and other things. So in humans, the excretory system consists of a pair of kidney, one pair of ureter, urinary bladder and a urethra. So what and all does our kidney consist, our human excretory system consists of a pair of kidney, okay, pair of kidney, and then one pair of ureter, urinary bladder and urethra. Okay, urethra. This is what our kidney has. A pair of kidney, one pair of ureter, urinary bladder, urethra. Now kidney, here they're talking about kidney. Okay, kidney is a reddish brown, okay, bean shaped structure situated between the levels of last thorax and third lumbar. So last thorax means the 12th thorax, okay, 12th thorax. It is extending from the 12th thorax up to the third lumbar. How many lumbars are there, students? 5 lumbars are there. So you label it L1, L2, L3, L4, no? So like that, T12 to L5. Okay, this is where your kidney is going to be present. So somewhere here. Okay, now each close to the dorsal inner wall of the abdominal cavity. Each kidney of an adult human measures around how much? It measures around 10 to 12 centimeter in length. So you can take that Natrak scale and measure, no, 10 centimeter scale you will get. 15 centimeter scale is decent. 15 centimeter scale you can search for 10 centimeter. Okay, 10 centimeter. So too much reflection is happening on the glass, sir. It's fine only, no? I'm just checking, okay? Sometimes it will be blur. Yeah. So around 10 to 12 centimeter in length, okay? 5 to, thank you. Okay, 5 to 7 centimeters in width, 2 to 3 centimeters in thickness. So wait, see, 10 to 12 centimeters in length, 5 to 7 in width, 2 to 3 centimeter in thickness, and an average weight of 120 to 170 grams. So 10 to 12 centimeter in length, okay. Width is how much? 5 to 7 centimeter in width. Then 2 to 3 centimeter in thickness. Thickness is less, okay. And 120 to 170 grams. Okay, weight. Weight is 120 to 170 grams. Towards the center of the inner concave surface of the kidney is a notch called hilum. So students, concave surface means this is the kidney. Okay, this portion is the concave surface. I used to hate so much to learn about concave and convex mirrors when I was in my 11th and 12th. How about you guys? Do you like concave and convex? I think I started from 10th because I did ICSE. So 10th it was there, 8th it was there. So every time it was there. So you people tell me how is your feeling about learning about convex and concave mirrors? Okay, this is the structure. So this is the concave, and this question has come, which is shocking because such small lines of NCERT they had picked and given as assertion reasoning, concave and convex. Okay, and many students got it confused. So towards the center of the inner concave surface of the kidney is a notch called a hilum. Okay, through which the ureter, blood vessels as well as the nerves enter. So what and all is going to enter? Ureter is going to exit. Okay, blood vessels are going to come in, and the nerves are also going to enter. Inner to the hilum is a broad funnel shaped structure which is called the renal pelvis with projections called calyces. So that you're going to see. So students, here look here, kidney has two zones. Okay, this is your kidney. This is, I think with pen I can draw a better kidney than on board. Okay, this is a beautiful kidney. This kidney has two zones. Okay, let's do the separation like this. Two zones are there like this. Okay, let's say this is your cortex. Outermost area is your cortex. Okay, cortex. And this is your inner area. Okay, innermost area, which is your, okay, this is the inner area, which is your medulla. Understood? Now wherever the dots are there, okay, wherever the dots are there, which you're seeing, okay, that is the cortex, and the inner portion is the medulla. So this portion, cortex, outer region, okay, outer region, this is medulla. Medulla, this is hilum, hilum through which the ureter is going to come out. Okay, ureter is going to come out, and the nerves are going to enter, and this portion is your pelvis. Okay, this is the calyces. Like the structures are going to be, this is going to be calyces. Now in the cortex region, okay, half of it, there is going to be the medullary pyramids. What is going to be there? Medullary pyramids are going to be there. This portion which you see. On the medullary pyramids is where your nephrons are going to be there. So here, the structure which you are seeing here is your medullary pyramids. Are you all getting it? Yes? Okay. Not bad, you people have all liked today without telling. Okay, projecting into calyces. So the cortex extends in. So the cortex extends in between. So here, yes. So students, look here. Not coffee, not tea also. It is for my throat. So it's actually ginger, honey, pudina. It's just a water where all of this is boiled. No, it's not warm water also. Warm water is still better. My expression, if seeing, you feel it is warm water. Okay, it is a decoction, we tell, no? Like that. Yeah. So students, look here. On top of the kidney, I am feeling that I will catch a fever. So I'm already taking precautions. Okay, so here on top, this is the adrenal gland. Okay, on top of the, yes, kashayam. Adrenal gland, then we have the renal artery. How come all of you are here? You people send message to each other, no? Hey, Gopika ma'am is live, let's go disturb her. Then we have renal vein, then you have kidney, dorsal aorta, okay, ureter, urinary bladder, urethra. Okay, here you have inferior vena cava, which is going to bring all the blood. Okay, then we have pelvis. Can you see the pelvis here? Pelvis extends and then ureter extends from it. Okay, and then you have medulla and then you have cortex. So this medullary pyramids is going to extend more towards the medulla, but it's going to start from the cortex. Okay, it's going to start from the cortex. Done. Yes or no? So here just write this. Okay, about your kidney. Okay, reddish brown color. Okay, reddish brown kidney. How much, students? 10 to 12 centimeter in length. Okay, 2 to 3 centimeter in thickness. This you have to know by heart. Again, match the following will come. Okay, 5 to 7 centimeter in width. I do a very simple maths to remember this. 10 by 2, 5. Okay, 10 divided by 2 is 5. Okay, then where is the location? Location is from the T12 last thorax to the third lumbar. Okay, third lumbar abdominal region. Okay, got it? Yes, yes, close to the dorsal wall. Close to the dorsal wall. Yes, close to the dorsal wall. Students, are you all excited for the Navratri series? It's starting tomorrow. Excited, huh? Which chapter do you think I'm taking? Weight we didn't write, no? 120 to 170 gram in weight. Excited. Okay. Now, students, kidney has two regions. Okay. Kidney has two regions. Cortex, medulla. Okay, cortex, medulla. Okay, done? All of you? Okay, now I'll teach you the diagram. Okay, 1 second. God knows, Tanuja. Oh, one looks okay. I had kept a diagram, but it's okay. Missing the diagram window. Now, students, there are some muscles, okay, that you have to study while learning your this one. First, we will start with kidney, and then we will slowly go to ureter, urinary bladder, etc. So, first, learn your kidney. Okay, this is the big kidney. It's bean shaped. Okay, bean shaped structure. Then here you have the cortex. Okay, cortex. Okay, what did I tell you was on the cortex? See, a very large portion is off to the cortex. Okay, medullary pyramid, and the same thing goes here. Okay, same thing goes here. Same thing goes here. Understood? Medullary pyramids. Now, on the medullary pyramids, who is going to be there? Like this structures, just draw this. Okay, on the medullary pyramids, your nephrons are going to be there. And who is nephrons? Nephrons are the structural and functional unit of your kidney. They are the people who are going to make the urine and everything, right? They are going to be the filtrating unit. So, this is the nephrons. Now, from here, see now, from the medulla, you are going to have extensions like this, okay? Obviously, extensions like this are going to come, like this and like this. Okay, they should be more close, but it's okay for us. Like this, just connect these. Okay, connect these, and those are going to be your renal pelvis. Okay, those are going to be your renal pelvis. Got it? And here, this renal papillae is there. Okay, this portion, students, there are going to be foldings which are called as calyces. What is that called? Is the color different? Yeah, too much green, no? My kidney vanished. There's an undo button. Gone. Are you people happy? Keep laughing at my diagram, okay? This is what God will give you people. They'll be like, you don't deserve to see this diagram also, so go home. See, now kidney became like it hasn't eaten food for like 1 year of life. Also ask sorry in the class for making my kidney go missing. Kidney. Okay, kidney ready. Okay, yeah, this has fever, so you should respect it. Done, students? Okay, done. Now let's label these parts. Okay, the parts. So this is your kidney, Okay, this is the medullary pyramid. Medullary pyramid. Okay, then this is your nephrons. Nephrons, they are the structural and functional unit of kidney. Okay, structural and functional unit of kidney. People shouldn't talk anything about the kidney, yaar. People study. Done. Chalo. This portion I told you, it's the renal pelvis. Okay, renal pelvis. Students, around here your calyces will come, but it's very difficult to draw. Okay, around there your calyces will come. Now calyces, understand two types are there: major, okay, major calyces and minor calyces. What is that? Major calyces and minor calyces. Okay, done? Yes or no, all of you? Okay, now medullary pyramids. Okay, now medullary pyramids, nephron is going to be there. From here the urine is going to come and it's going to go from the ureter to the urinary bladder. Okay, now students, here your artery and vein entry is going to be there near the hilum. Okay, the vein is going to enter with the deoxygenated, no, with the blood. Okay, with the blood. Again, to carry also we will have one more person. You can draw it this way or you can just draw it this way. Okay, like this. Got it? Now students, one question students usually have is, ma'am, is deoxygenated blood rich in waste? No students, remember, deoxygenated blood is in only terms with carbon dioxide. Carbon dioxide is more, we will call it deoxygenated blood. Imagine carbon dioxide is less. Okay. Kichi, Naman, your name sounds like that dal khichdi. Okay. So yeah. So this deoxygenated, it's very easy for people to just comment or anything. No? Okay, so deoxygenated is like carbon dioxide. Carbon dioxide is more, and oxygenated. Okay, oxygenated means oxygen is more. It does not mean that they are toxic. Okay, they have toxicity or whatsoever. Okay, so deoxygenated means they are less toxic. Okay, they do not have toxicity in their blood. That means the excretory substance, no, urea is not there. This one will be more toxic. Toxic. Yes, got it? Only Tanuja got it. Clear? And students? Chal. Okay. Now here we have to learn about the next two organs. Okay. So here, I hope you people understood. Ureter. This ureter has some muscles which I have taught you in structural organization. The muscle, okay, animal tissues when we learn, okay, animal tissues when we learn about compound and simple. So under compound, students, okay, under compound there is two types. Okay, in that one type is called as your transitional epithelium. What epithelium? Transitional epithelium. Transitional epithelium. This transitional epithelium is going to be present on the ureter. Okay, it's going to be present on the ureter. Yes. So students, let's assume this one. Okay, the green color one is the transitional epithelium. It's too difficult to draw actually with the pen. Okay, transitional epithelium. On top of that, you are going to have the connective, muscular and connective tissue. Okay, there's one more layer which is muscular. I'm not able to draw here. This is going to be your muscular layer, and on top of the muscular layer, you're going to have a connective. Okay, connected. It's someone else. Got it? This is going to be your muscular limb. Now what are they going to do, student? They are going to do something called as peristaltic movement. Okay, you people did not answer which chapter I'm taking in Navaratri series. Did you people guess? People who are guessing. Peristaltic movement. That means that movement, no, that has to happen, and that movement is going to help in the passage of urine up to the urinary bladder. Now, same thing you can see it in the urinary bladder, students, but urinary bladder you have to learn some more things. Okay. Now let's say this is the urinary bladder. Same muscle arrangement is there, but the second muscle, that is the detrusor muscle, is there. Surprise was one surprise was Navaratri series. One more surprise is coming. Okay, on the way. You have to wait maybe up to the second week. Okay, this is the muscle. Okay, let's say blue is the transitional muscle, pink is the muscular muscle, and upper to that we have the connective muscle. Okay, connective. Now, students, the catch here is, okay, the catch here is this muscle, no, muscular one is going to be called as the detrusor muscle. It's a type of muscle that is going to help in the contraction. So when you're like, let's say when you're feeling full, okay, or you feel like, okay, I need to use the washroom, what is going to happen? This urinary bladder is going to contract. Okay, urinary bladder is going to contract, and this urinary bladder is going to send signal to your brain telling that, wait, this person have to use the, or this person has to expel the urine out, or micturition should happen. That is when your muscles are going to work. So that is called as detrusor muscle. Now, students, here remember the ureter is going to come like this, right? One ureter from here, one more ureter from here, okay, and there is a urethra, okay, urethral opening here. Now this three openings together we can call as trigon of bladder. What can we call? Trigon of bladder. Understood? Trigon of bladder. Tri means three. Trigon of bladder means three openings together. What are the openings, students? Openings are nothing but very easy for you to remember. Ureter, two openings of the ureter plus one opening of the urethra. Okay, one opening of the urethra. Two openings of the ureter, one opening of the urethra. So this one. Now in the end, okay, in the tip of this, there are two muscles, okay, two openings called as sphincter muscles. Not opening, two muscles called as sphincter muscle. One is the external sphincter muscle. Okay, I don't know if it's visible to all of you because I've used so many colors. Okay, this is the external, sorry, this is the internal sphincter muscle, and this is the external. Okay, now students, internal sphincter, okay, internal sphincter muscle is not in your control. It is involuntary. Involuntary. That means now if you imagine that you want to control it, okay, now how many of you have experienced this? You're going to a shop nearby your home, okay, let's say you have some distance to go, and like till that time you're fine, you went to the shop, you bought something, suddenly you'll start feeling like, okay, I have to use the washroom. Suddenly at one point you'll feel like, oh my god, I think I'm gonna micturate. Micturation is a process of expulsion, okay, so you're gonna be like, oh my god, I think this is gonna expel out now and I'm gonna get embarrassed in front of everyone. Okay, why you're having that feeling is because your urinary bladder starts getting full, and up to 800 ml your urinary bladder will be able to hold. Usually it's 700 to 800, okay, 700 to 800 ml. So when you were going to the shop, let's say your urinary bladder was around 400 ml full, and as you were walking, it reached 500 to 600, 600 to 700, and by the time it is 700, you will not be able to have a control of your internal sphincter muscles. Okay, internal sphincter muscles. So this internal sphincter muscles would have already opened. Now what can you control? Now how many of you have felt this, that you see the washroom, like you are near the washroom and you feel like you would have already done micturition on just entry to the washroom? That also happens because of the feeling that our brain has sent the signal that okay, this person is in a place where their micturiation should happen, okay, or urination should happen. That is the reason by then your external sphincter muscle also, like, it would have gone out of your control, because again, why? Urinary bladder is not able to hold any more urine. Okay, so students, it's very simple. Here there is a stretch receptor. Okay, here there is a stretch receptor. This stretch receptor will tell the brain that, you know what, urinary bladder is getting full, urinary bladder is getting full, please quickly expel it out. So internal sphincter muscle already would have been open, it is not in your control. External sphincter muscle is in your control. Okay, understood this? External sphincter muscle is in your control. That is voluntary too. Voluntary. Okay. Voluntary. Say voluntary. Done. Chal. Okay, this is clear. Now, some more structures of your kidney. Okay. Now students, medullary pyramids are as renal columns called columns of Bertini. Each kidney has nearly 1 million complex tubular structures called nephrons, and nephrons are the functional units of each nephron. Each nephron has two parts. So here they are telling there is a column of Bertini. Okay, column of Bertini. Actually, they say that column of Bertini is an extension of the cortex. If you have noticed, okay, between the medullary pyramids there are spaces. Yes or no? Between the medullary pyramids there are spaces, and those portions are the cortex. So cortex is extending downwards to the medulla, and that is called as column of Bertini. Understood? Okay, now students, they are telling that nephron, okay, nephron is the structural, or let's say functional unit mainly. Okay, nephron is the functional unit of kidney. Functional unit of kidney. Now this nephron has two portions. Okay, nephron has what? It has two portions. One is called, see what is one called? Glomerulus. Okay, glomerulus. And one more is called renal tubule. Renal tubule. So here glomerulus is going to be in the Bowman's capsule. All of you know, no, a cup-shaped structure, Bowman's capsule. So if I have to draw glomerulus, okay, I'm a very big artist, so I will draw. Okay, here you go. Okay, blood is this. This is your glomerulus. A network of capillaries. A network of capillaries. Guys, don't make me so famous, then they'll ask my diagram. Okay, then I'll have to sell it. It's going to become too difficult. Okay, too much money I'm going to have, then I will not know what to do. So don't please make me famous. Okay, yes, understood. See, that is the glomerulus. Okay, that is this network of capillaries. Okay, network of capillaries is called your glomerulus. And students, renal tubule has three portions called as PCT. Okay, loop of Henle, and DCT, distal convoluted tubule, proximal convoluted tubule, and loop of Henle. This is a network of capillaries. Understood? Now let's read further. Glomerulus and renal, okay, and the renal tubule. Glomerulus is a tuft of capillaries. That means a mesh of capillaries formed by the afferent. So the arteries that is going inside, no, this is called as afferent, and the arteries that is going outside is called efferent. Okay, so a fine branch of renal arteries. Blood from the glomerulus is carried away by the efferent arterioles. The renal tubule begins with a double-walled cup-like structure called the Bowman's capsule, which encloses the glomerulus. Glomerulus along with Bowman's capsule together is called as the Malpighian corpuscles. What is it called? Malpighian corpuscles. Oh my god, they'll pakka make us buy the milk, no? They're like, how many ads are they telling? Malpighian tubules. Are you also getting the same ad? All of us are getting that. Okay, now the tubule continues to further gets highly convoluted into PCT, DCT and loop of end. Now ready? Okay. Now what if we are lactose intolerant? I think they will not allow us to live life. Now students, I forgot to tell you one thing. In case the stretch receptor is not working, then there is a condition called incontinence. That means it happens in old age people. They will not get to know that the urinary bladder got full. Okay, so there will be leak of urine. Got it? Leak of urine. Now students, flow of blood. Okay, flow of blood I'll teach you on content. See, now there are some portions that I have to tell you. Hilum, notch, okay, at the concave side. Then calyces, projections in the funnel shaped, okay, funnel shaped spaces. Capsule, outer layer, okay. Capsule I did not tell you, no, while teaching you kidney. Capsule, renal capsule, students, here, that's the kidney. Here, this outside, no, there is a protective layer called as the renal capsule. Okay, renal capsule. Now for board students, only renal capsule word you have to study. For NEET students, I have already taught you capsule has 3 portions, right? Okay, or if you remember, fasciculi, your adipose tissue that makes your entire renal envelope, right? Then 2 zones are there, medulla and cortex. Column of Bertini is actually cortex extending between. So this portion, no, this portion is actually column of Bertini. Here where I have drawn the arrow mark. Okay, yes, renal envelope, very good. This is called as column of Bertini. Why I am calling this column of Bertini? Because where the cortex is extending towards the medullary pyramid, okay, medulla is called as column of Bertini. Got it, students? The nephrons, 1 million per kidney is that. So let's go further. Now, I have to teach you so many things. I'm thinking what to write now. Okay, chalo. We will write this only. Okay, what is the portion? Okay, we will just draw the whole this one. Okay, I think I'll draw it with white and then later we will extend it. So students, this is how it's going to look like. Okay, this is your Bowman's capsule. Okay, this is the Bowman's capsule. It's going to extend. Okay, and it is going to get highly convoluted. Okay, highly convoluted. Like this, convolutions are going to happen. Then it is going to loop off Henle. Then it is going to extend. Okay, then again it is going to and this. Okay, this is how convolutions usually happen. Actually, it's a very complicated structure. If you actually search for the nephron real structure, you will not even be able to understand which is DCT, which is PCT. Nothing you will understand. Okay, nothing you will understand because it's very convoluted, very convoluted. Now students, one question I want to ask you: which is the part of the nephron, which is not the part of the nephron? Tell me which is not the part of the nephron. Which is not a part of nephron? Keep thinking. Okay, I'll hit you people. It's collecting duct. Collecting duct is not a part of the nephron, a single nephron. Why? Because multiple nephrons open into it. Okay, multiple nephrons open into it, so it's not a portion. Okay, it's not a portion. Understood? So this is the structure. Okay, yes, collecting ducts, very good. Because see, collecting ducts, see, I have drawn different. So here one nephron is coming, here one nephron is coming, here one nephron is coming. So it's a different structure. Okay, now student, this portion I have to erase. Oh god, all half it's not going. Okay, wait 1 second. This loop of Henle. I'm just convoluting it a lot so that you understand like how much convolutions can be there. Students, I had drawn the very good diagram in NEET English. I don't know if you people remember. Remember, we drew a very good diagram during the RNA series. Done. Okay, this is your tubes. Okay, this is your tubes. So I have the flashcards with me, as we already saw. So these are the flashcards. Okay, now these flashcards have the cutie diagrams of your NCERT. Okay, they're very pretty, very pretty they are. So I'm using the flashcards to give you some additional information anytime like we miss some information. Okay, chalo, let's draw the glomerulus. So this is the glomerulus, this is the afferent arterioles that I've drawn. Okay, and it's extending like this. It will definitely stretch. Okay, it will definitely stretch like this. This blood vessel, students, there's no pathway as such. It is going to go like this, but here it is going to go exactly below the loop of Henle, extend towards it like this. Okay, and connect somewhere like this. Okay, done. This is your blood. Should I have drawn it in yellow? I mean red, sorry, yellow I'm telling. By then ad will get over. So blood is done. Okay, blood vessels are done. Now listen here. Okay, now we know two things. Nephron. Okay, nephron has two portions, glomerulus and the renal tubules. Okay, now renal tubules, student, this is going to be your Bowman's capsule. Okay, Bowman's capsule. It's a cup-shaped structure that is holding the glomerulus. This portion is going to be your efferent arterioles, A. Okay, this is going to be efferent. That is, one is going to bring the blood to the kidney and one is going to take the blood away from the kidney. Okay, now this portion, okay, this portion is going to be the PCT. That is proximal convoluted tubule. Okay, this portion, this full extending downwards U-shaped structure. This U-shaped structure is your loop of Henle. Okay, loop of Henle. Then this portion is your distal convoluted. Distal convoluted. Okay, distal convoluted, that means DCT. Distal convoluted, and this is the collecting duct. This is the collecting duct. Clear, all of you? Yes or no? Okay, now so this portion, okay, this portion, this actually some places I should have drawn in blue, but I haven't drawn some places in blue. So it's okay, just understand the blood vessels, some portions are going to be blue. Okay, just increase the thickness and draw. Okay, this portions, some of it is going to be blue. Okay, and they're going to connect like this. They're going to connect from here to here. Okay, now students, these are called as, okay, these are called as peritubular capillaries. What are these blood vessels called as? Peritubular capillaries. Got it? Peritubular capillaries. Now peritubular capillaries are going to extend again, okay, to form the vasa recta. Vasa recta. That means a small division of peritubular cavity, capillary is called as your vasa recta. Clear? Now, in between this peritubular capillary, students, your renal vein is going to have an opening. Okay, a renal vein is going to have an opening here. Somewhere at this portion, the renal vein will have an opening to the peritubular capillaries. This way only blood is going to come in, go out, all that is going to happen because of this. Clear? Done? Okay, and here your arteries, okay, renal arteries is going to have a connection like this. Not like this exactly, but yeah, I'm just showing it like this. Okay, I'll label Bowman's capsule that side. Okay, now let's assume here renal arteries are extended. Okay, they're gonna have connections like this. So the blood is gonna come So you understood, no? Who is going to go for oxygenation? It is going to be oxygenated blood. Your oxygenated blood is going to have more toxic substance. Okay, your oxygenated blood is going to have more toxic substance. So deoxygenated blood is going to be having less toxic substance, or more, let's say, generally less. Okay, now here, this is the renal vein. Any doubts here? Diagram is clear? Diagram is clear? Very colorful diagram, no? All the portions are clear or not? Tell me. Now this loop of Henle, students, this loop of Henle has two portions. Okay, one is called as the ascending limb. Okay, ascending limb, very important for you, because here you will learn more about ascending and descending limb when you're learning about urine formation. Okay, descending limb, ascending limb and descending limb. Got it? Done. Okay, hi sweetie. Yes, this is your entire thing. Now the diagram here, look at this diagram. This was the diagram I drew before. See, pyramids, can you see? Pyramids are actually small. They're starting from the cortex and they're extending into the medulla. Okay, from here, can you see this small portion? These are your calyces. Okay, calyx. Can you see here? Actually, there are foldings here, students, like this, like this, like this. Major calyx and minor calyx, we tell. Then you have renal column. Column is here, column of Bertini. The cortex is extending towards the medulla because of it. Then you have renal artery, renal vein, renal pelvis. Pelvis is this large portion. The urine will be collected here and it will go back. It will come to the ureter. Okay, ureter, renal capsule. Capsule is the covering. Okay, covering of the kidney, which is going to protect the kidney. Okay, which is going to protect the kidney. Then you have cortex and medulla. Clear this diagram? So if you want, I'll write the definitions in the flashcard here. Okay, so you can use this later, or you can write it now only. Hilum is a notch. Okay, present on what surface? Present on the concave end. Concave side of the kidney. Okay, concave side of the kidney. Okay. That now concave side of the kidney. Okay, this hilum. So students, to the hilum you can see arteries, veins are entering, nerves are entering, ureter is coming out. Okay, next is going to be calyces. Okay, or you can write calyx. What are they written? Projections. Okay, projections in funnel. Projections in funnel shaped space. Okay, this is lines of NCERT. You'll get this PDF on the Telegram, dear. Biology by Gopika. Usually all the notes are shared there before only. Oh, yesterday's notes I have to share it to you guys, no? Breathing and exchange. I'll share that, students. I forgot. Okay, 2 zones are there, done. Now columns of Bertini also I'll write. Team CEO asked. Maybe you blocked that name. Okay, medullary pyramid. Yes, okay. Chalo, clear? Done? Okay, students, we are going to take a break. Okay, this is the diagram I drew. After this, we'll take a break. Okay, 7:30 we'll take a break. So look here. Efferent arterioles, the blood is going out. Okay, going out. Afferent arterioles, blood is coming in. Okay, this is connected to the renal artery. Renal artery. This is connected. No. This one. No. Okay. Now proximal convoluted tubule, the first part of the renal tubule. Okay. Then Bowman's capsule. Yes, Bowman's capsule. Then students descending limb. Students descending is the upper one. Okay, ascending is the nature, the side one. So descending loop, ascending loop. Vasa recta is clear, no? Collecting duct. Distal convoluted tubule. Okay. Now remember this. Okay, remember this. Peritubular capillaries. Okay. Peritubular capillaries is going to extend, or like what to tell, a small portion is going to form the vasa recta. Okay, and this vasa recta is going to help, vasa recta plus loop of Henle is going to help in the counter current mechanism. Counter current mechanism. So sometimes we don't teach extra, it's just that to make a connection, because NCERT sometimes just mentions the words. Now you will not know what is vasa recta, so that is why we teachers tell a little extra. Now peritubular capillaries is not mentioned there as such, so when we tell that, you will be able to connect it. Understood? You will be able to connect it. Clear? Okay, we'll come back and do this. Okay, yeah. Yes, sure. See you guys also after break. After that, I'll teach you guys mechanisms. Okay, then that's all for today's class. Hello students, I hope I'm audible. There was a slight network issue here. It is raining here very heavily. That's the reason I got a little late. I hope now the network is fixed. Okay, I think yes, we are live. Okay. Voice is fine, right? Okay, I have to disconnect the Wi-Fi and connect the Wi-Fi. All the task was there. Now it should be fine. Okay, so we stopped up to our kidney. Now here there are some lines given by NCERT, so let's go through that as well. See, a hairpin shaped Hendy's loop is next part of the tubule, which has a descending and an ascending limb. The ascending limb continues as another highly coiled tubular region called as distal convoluted tubule. The DCT of many nephrons open into a straight tube called the collecting duct, many of which converge and open into the renal pelvis through the medullary pyramid. So can you relate to it, how they're opening? So this is your medullary pyramids. Okay, this is the medullary pyramids. Okay, this one. Students, anytime network goes, just let me know, okay, because I won't get to know until the video, like here it stops. Okay, so here this is your Bowman's capsule. Okay, this is your PCT, this is the loop of Henle, this is your DCT, and this is your collecting duct. Okay, collecting duct. Now what are they telling? They are telling that the DCT of many nephrons open into a straight tube called the collecting duct, many of which converge and open into the renal pelvis, okay, through the medullary pyramid. So through the medullary pyramid, these collecting ducts are going to open into the renal pelvis. This portion, no? This portion is the renal pelvis. Okay, like this. It extends, no? To this portion, the urine is going to come. From there, it is going to go into the ureter. Okay, ureter to the urinary bladder. Clear? Okay, chalo. Now the Malpighian corpuscles PCT and DCT of nephrons are situated in the cortical region of the kidney, whereas the loop of Henle dips into the medulla. In majority of the nephrons, the loop of Henle is too short and extends only very little into the medulla. Such nephrons are called cortical nephrons. So students, there are two types of nephrons, okay? There are two types of nephrons. That is, depending on the basis of what they'll ask you question, no? So nephrons, okay, nephrons are of two types, 1 and 2. On basis of what? On the basis of loop of Henle, okay, on the basis of size of loop of Henle. Loop of Henle. Have you guys ever taken a fast? Have you ever done fasting? Fasting? Yes, tell me in the chat box. Two nephrons are there, cortical nephrons and juxtamedullary nephrons. Yes, ma'am, you people have done. So students, when you're fasting, no, the nephron that is going to work in your body is called as juxtamedullary nephron. Ma'am, why so? Because I'll tell you, cortical nephron in your body is around 85%. Okay, so out of 1 million nephrons that you have, 85% is your cortical nephron and 15% is your juxtamedullary nephron. This cortical nephron, to be specific, this nephron has a short loop of Henle. Okay, short loop of Henle, and they have a negligible or hardly any vasa recta. Okay, they have no vasa recta, we can tell, or very less amount, because loop of Henle is not there, so vasa recta is absent or negligible. Okay, negligible. Now students, these people work during the normal condition. Okay, normal condition. Now if you know countercurrent mechanism, you people should know that to regulate the osmolarity, we do countercurrent mechanism. Okay. Oh, okay. So to do your countercurrent mechanism, we need vasa recta and loop of Henle. And why do we do countercurrent mechanism? To make sure that the osmolarity is maintained. Now, why osmolarity has to be maintained? So that, you know, a person who's not drinking enough water, that's when I realized I haven't drank enough water. Okay. So students, so imagine a person haven't drank enough water the whole day, or they were on a fast, and to make sure that that person does not, go through anything, okay, go through anything, like the person is happy or the person is able to do other activities, we need to make sure that the body should be able to do that. So for that we do this counter current mechanism, okay, for that we do this counter current mechanism. So here this has long loop of Henle. Okay, if long loop of Henle is there, that means it is extending more to the medulla. Okay, it is extending, okay, to the medulla. Then here you can tell vasa recta is present. Okay, vasa recta is present. Got it? Yes or no? Is present. Chalo. Then here students, this is going to work during the stress condition. Okay, this is going to work during the stress condition. Done? Clear? Yes or no? Okay, now this is going to work during stress condition. Why I am telling? Because this will have a long loop of Henle, and let me tell you, this is how the structure is going to look. Okay, this is how your cortical nephron is going to look. This way. Okay, very short loop of Henle. Okay, DCT collecting duct. Okay, here it is going to look ulta, right? This is your Bowman's capsule. This is your structure, long loop of Henle, like this, and your collecting duct. Understood? Yes. Now here, okay, remember this. Here there is going to be, okay, vasa recta, okay, long vasa recta. Here there can be no vasa recta. Clear? This is up. So juxtaglomerular nephron is going to work during the stress condition because it is going to help in counter current mechanism. Counter current mechanism. I'm doing good, Sahil. How are you doing? Okay, done. Now students, that is where I'm stopping here. Okay, that is urine formation I'll do in the next class. Before that we are going to learn some mechanisms. Okay, before that we are going to learn some mechanisms. Oh, how much are you spamming for that? I am good. I hope you are also doing good. So we're going to do regulation of the kidney function. What happened to my Avas, Mr. Sahil the Great? Okay, chalo. He doesn't seem to do well, students. Okay, I am doing good, but you don't seem to do well. Okay, obviously I'm sitting in a closed room. It's going to echo. So a little common sense. Okay. That is because of the mic, dear. I'm doing it at a small room, so it's echoing. Understood? That's why it's coming in that. Can't do anything. Okay. Chalo. Ready? Ras mechanism. Chalo. Ready? Ras mechanism. Yes. So students, first full form of Ras mechanism. Renin, angiotensin, aldosterone, okay, system. This is your Ras mechanism, okay, the Ras mechanism. Yes, all of you know Ras mechanism. Students, before Ras mechanism, I have to teach you JGA mechanism, no? Otherwise, how will you know Ras mechanism? How will you know Ras mechanism? Echo is there? Wait 1 second. How will you know Ras mechanism? Echo is there? 1 second. Wait 1 second. Is it better now? Then what happened here? The Sahil put eyes on our voice, students. What happened here? The Sahil put eyes on. Yeah, now it's fine, no, Tanuja? Just let me know. Now my phone only is echoing. Okay, renin angiotensin mechanism. Oh God, I think before the network goes, I should be able to do this. Better, Tanuja? Now fine. Okay. Yes, I'll talk softly so it doesn't echo. Friends, JGA apparatus. Okay, this is the juxta medullary. Okay, juxta medullary apparatus. I think this Rathas he doesn't have Oxford dictionary. He has his own dictionary. Have you seen how he types the words? He'll change the first letter. Okay, he'll change the first letter. Students, I love my voice. Okay, I love my voice. It's one of the things my teachers used to tell when I was in school. Okay, they used to be like, you'll always become a speaker. Okay, or you'll become an orator, or you'll become something else. Okay, and finally I ended up being a teacher. So what used to happen is when I used to take presentations, okay, when my like when I was in PU, I used to do presentations. When I was in my degree, I used to do presentation. When I was in my masters, I used to do presentation. So what used to happen is all my friends will be sleeping. Okay, like previously someone would have come and spoke, and the moment I come to the class, okay, I get in and I start teaching, no, or I start explaining, my voice used to be like so much so loud it used to be, and there's clarity in my voice, like it's very loud but there's clarity in my voice. So everyone used to wake up and sit like this. So then they'll be like, please don't do presentations anymore, we can't sleep in with your voice. So I feel I love my voice. Okay, yeah, it has a lot of bass. It's not like that key voice. Okay, it's not like that bird ka sound that will be so annoying, but I have a more bass in my voice. Okay, because my larynx, I think larynx is a little enlarged, so my voice is why I'm a teacher today, I feel, because my voice is makes a lot of students. I've got a lot of compliments. Usually people will tell, ma'am, it's very like, what to tell, bass is more and all of that, but many students have complimented for my voice, so I really love my voice. So when people tell something, so see, if you love something about yourself, I don't think anyone else is having the rights to talk about it. So always be like this. Once you have a confidence about one of the things that other people might think that, oh, it's her weakness or it's his weakness, but you feel confident, then there is no going back, students. I've always felt this. Like, see, if we are like, say you have an insecurity, okay, that I am chubby, or I am that, I am this, I'm not like a model figure, all that. You might have insecurities at this age because you're going through adolescence. Okay, since you're going through adolescence and it's normal for you to feel such things, what is going to happen? You're like, you will start comparing yourself with other people, you'll start comparing yourself with a celebrity and all of that. Okay, so but just realize that once you start loving yourself in the mirror, no one is going to stop you. I've learned that. See, no one tells all of this, in home, or parents doesn't tell all this and give you, but just understand this. Okay, I'm like your sister, elder sister. You can just listen this from me. You love yourself. When someone tells something, okay, so once you tell something, like let's say someone tells, okay, that ma'am, your voice is like this, or ma'am, or let's say something else, okay, you tell that, no, I really love my voice, automatically they will also start loving your voice. They will also start loving whatever things that you have. Okay, yes, clear? So I've given you a model education class, very free education class. Okay, you'll never get it for free. I don't like usually giving motivation, but if it comes in a flow, then it has to come out. Okay, if it comes, then you have to listen to it. It usually doesn't come very easily. If it comes, then you don't have a choice, you have to listen to it. So yeah, learn this lesson today, that love yourself, and then people will love you automatically. And students, even in this exam journey, competitive exam journey, the one thing that you need to love is yourself more. That is the reason why that student who got Air 1, he committed suicide. Okay, he was doing his radiology, other things he was doing with his life, but he didn't love himself enough. Maybe he's going through a lot of things, but you should love yourself more than a competitive exam, more than a course, more than a job. You should love yourself. You should not be obsessed. Obsession is a different thing. All the self-obsession, people will become psychopaths. Not obsession, but you should like yourself that, okay, my life is more important than this competitive exam. My life is more important than this job. My life is more important than this toxic environment that I'm staying. Okay? Moral of the story is, you are not fit to listen to moral of the story. Okay, that's the moral of the story. Don't worry, this is my regular student, so I can scold him, and he's not going to even have little feelings. Also, he'll not have. I want him to have feelings, but he doesn't have. Yes, life is very important. Exactly. Only Sahil understood. Okay, they're all monkeys. From dinosaur time, they are here, so they have lost that feelings also. Okay, done. Ready? See, I told you, dinosaurs' behavior. Food is more important. See, that's why dinosaurs went off. Okay, you are the reason. They left you and went. Tell the reality to everyone. Okay, so yes, students. So this is about a 65 million years ago story. One student got left here. Okay, one dinosaur got left here. That is Rathis. For him, food, bigile food tha mukyam. Like that, this fellow says. I'm telling moral of the story means you should tell, wow, ma'am, what a psychology. Wow, I started loving myself. No. What to do? Wrong students, I gave lecture. Okay, listen, look here. Now this is your Bowman's capsule. Okay, I have made it ulta. Okay, ulta. So how is your Bowman's capsule? Like this. Okay, Bowman's capsule is like this. Okay, this is your glomerulus. Yes or no? This is your glomerulus. Now students, DCT will come very close to this. So if I do not draw it ulta, you will not understand it. Okay. So that's why I've drawn it ulta. Now understand, whenever there is going to be an issue. There are only those two people. Okay. Students, here if I draw, you'll be able to see, no? Okay, this is your macula densa. Okay, so what is this juxtaglomerular apparatus? It's very simple. See students, whenever there is a problem with GF, okay, don't think your girlfriend, not your girlfriend, it is glomerular filtrate. Okay, glomerular filtrate. Where is glomerular filtrate coming? It's coming from the sea. For urine formation to happen, there is glomerular filtration that is going to happen. So there is going to be a formation of filtrate. Okay, formation of filtrate. Now that filtrate, okay, okay, this glomerular filtrate, if there is a problem with glomerular filtrate, like example, water is low, okay, or blood volume is low, okay, blood volume is low, or BP is high, that means sodium levels are high. Such problems can normally come, yes or no, when you're making tea every day. Okay, when you're making tea every day the tea will not taste same, no? Some days chai will, the leaves will be more, sometimes sugar will be more, different things can happen. So whenever there is a problem with the glomerular filtrate, there should be one mechanism that will recognize it. So that mechanism is called as juxtaglomerular apparatus. Now this portion which you're seeing here is your Bowman's capsule. Okay, Bowman's capsule, and what you're seeing down is your glomerulus, and near to that only you're going to see your DCT, that is distal convoluted tubule. So in DCT there are some specialized cells called macula densa. What is it called? Macula densa. This macula densa, okay, this macula densa will recognize that the GF has some problem. Okay, the GF has any problem. Got it? Now this glomerular filtrate, if they have any problem, who is going to be released? This afferent arterioles, okay, the afferent arterioles, this is afferent, no? Afferent arterioles, some of the cells, some of the smooth cells, smooth muscles, you can tell, some of the smooth muscles will modify themselves, okay, smooth muscles will modify themselves to form JG cells. What cells? JG cells, that is juxtaglomerular cells. And this JG cells will release renin to fix the issue. JG cells will release whom? Renin. Understood? I cannot explain in Hindi. Okay, this is English. I mean, I can talk only English. Okay, so students, what did you understand so far? Glomerular filtrate has some problem. That is, blood volume is high, water is high, blah blah blah. Some problem is there. Now, when this is flowing through the DCT, will have a special type of cell called as macular densa. We can also call this as sensor cells. Okay, sensor cells. Now, this sensor cells will understand that GF has some problem, and they will tell afferent arterioles, they will signal afferent arterioles. Okay, okay, okay. So this macular densa will inform afferent arterioles that, okay, afferent arterioles that, you know what, there is a problem with the GFR, so we have to do something. So then afferent arterioles will be like, don't worry, my smooth muscles are going to get modified to JG cells and they are going to release renin. Is this clear so far? Yes, all of you? Okay, it's okay, relax, don't be furious. Okay, all of you calm down in the chat box. Okay, renin, angiotensin, aldosterone system. Okay, हमें हिंदी नहीं आती. Okay, so please don't ask us to talk in Hindi. Okay, ready students? Renin. Okay, this JG cells released renin. What did this JG cells release? They release renin. But students, there is a problem. Renin cannot do much. Renin has a problem. What happened? This renin, okay, renin will be like, okay, I cannot directly fix the issues in the GF. So what I am going to do, I am going to activate angiotensinogen. Angiotensinogen. Okay, angiotensinogen where? Okay, see, this angiotensinogen, students, is present in the plasma. So this is your blood vessel. Okay, this angiotensinogen is present in the plasma, and this angiotensinogen will get converted to angiotensin 1 with the help of renin. Angiotensin 1 with the help of renin. Now, we'll ask angiotensin 1, can you fix all of the issues? Angiotensin 1 will be like, no, I cannot fix the issues, so I will make angiotensin 2. Now, to form angiotensin 2, we are going to take one more person's help. That help is going to be with the help of whose? Lungs help we are going to take. Lungs only, no students? Check once. Liver done, lungs, yeah. Okay, with the help of lungs, we are going to take the help and get ACE enzyme. Okay, what enzyme? ACE. He knows English, that's why he's here, no dear. What are you people telling? So with the help of lungs, we will get ACE, that is nothing but angiotensin converting enzyme. Okay, angiotensin converting enzyme. Clear? Yes. Okay, now we got angiotensin 2. So we'll ask angiotensin. Angiotensin 2, can you fix the issues? Now, what is the issues? What are the problems that we faced? The problems we faced was BP was low. Okay, blood volume was low. Blood volume was low. Water was low. Okay, GFR rate was low. These are the issues or the problems that we faced. Clear, all of you? Now angiotensin 2 will be like, don't worry, I am going to fix all the issues one by one. Okay, so angiotensin first will release aldosterone. First will release what? Aldosterone. Tell me students, aldosterone is released by whom? Tell me, all of you. Tell. Aldosterone is released by who? Adrenal gland, thyroid gland. Okay, by the time you people tell, I'll write here. So aldosterone will do conditional, okay, conditional reabsorption, conditional reabsorption of sodium. So when you do conditional reabsorption of sodium, automatically BP will get fixed. Very good, adrenal gland. BP will get fixed. Okay. Next, angiotensin will be like, what is the next issue? Then we will tell the angiotensin that, you know, blood volume is low. Okay. Or water is low, we'll tell. So angiotensin 2 will be like, don't worry, we will do reabsorption of water. We will do reabsorption of H2. So these two issues will be fixed. Then we will tell GFR is low. That means the blood which is coming in is low. So then they who will your angiotensin will tell, don't worry, we will do vasoconstriction. Okay, or vasoconstriction of whom? We will do vasoconstriction of the efferent arterioles. So the blood which is going out will be reduced. Is this clear? It's a very, very important mechanism. Okay, vasoconstriction of the efferent arterioles. Done. So students, what is RAS mechanism? Okay, what is RAS mechanism? It's very important for you to know. Students, RAS mechanism is renin angiotensin aldosterone mechanism. This happens when there is a problem with the GFR. Okay, so we already understood how renin is released. Renin is released by your juxtaglomerular apparatus. Okay, so JG cells will release renin. Now renin will be like, okay, I will fix the issues, but renin cannot directly fix the issues. So in the plasma, okay, in the plasma there is going to be angiotensinogen which is formed by your liver. Okay, angiotensinogen is actually formed by your liver. Okay, liver. So angiotensinogen will be converted to angiotensin 1. Angiotensin 1 will be converted by angiotensin 2. Okay, to angiotensin 2. So tensinogen will be converted to angiotensin 1. Angiotensin 1 will be converted to angiotensin 2 with the help of ACE enzyme, that is angiotensin converting enzyme, which will be taken from the lungs. Now angiotensin 2 will be like, guys, don't worry, I'm going to fix all of your issues, and it will fix all the 4 issues. What are the issues? BP was low, or the sodium content was low, blood volume was low, water was low, GFR was low. Okay, so they will be like, we will do conditional reabsorption of sodium. This way your BP will get fixed. Second thing, by the release of aldosterone. Okay, second thing what it's going to do is reabsorption of water. It will be like, don't worry, we will do reabsorption of water. Last thing they will do is vasoconstriction of the efferent arterioles. That means, students, see, okay, just look, go here. Okay, go here. Now what they have done is, see, this is the Bowman's capsule. Okay, this is the Bowman's capsule. Yes or no? This is the Bowman's capsule. Now when efferent arteriole is coming, okay, with the blood, they're telling that the GFR is low. That means the blood that is coming in is low and the blood that is going out is more. So here, this is the tube, no? This is the tube. So they will not do vasodilation. Instead, they can do vasoconstriction. So let's say this is the length of the pipe. Here, what they're going to do is, students, here they will reduce it. Okay, here they will reduce it. So here you can feel, no, vasoconstriction has happened, yes or no? So here they will do vasoconstriction. Okay, this way what will happen? The blood that is going out will reduce. Okay, the blood that is going out is going to reduce. Okay, and the blood that is going in will be normal as usual, and the GFR will become normal. GFR means glomerular filtrate rate. Is this clear to all of you? Yes or no? Chalo. Now the next mechanism, okay, next mechanism is called as ADH mechanism. ADH mechanism. Actually, students, here in the flashcard they have given one, but it's okay, we will just write this. Okay. Okay, never mind. Okay, this is only fine. Okay, this also is there, it's good only, but then no need. Here, okay, ADH, first you need to know the full form. Anti-diuretic hormone. Anti-diuretic hormone, other name is vasopressin. Now all my students have studied chemical coordination with me. You already know that vasopressin is released by the hypothalamus. It is stored in the posterior lobe. Yes or no? It is stored in the posterior lobe. So students, what happens? Whenever there is loss of fluids, okay, loss of fluids from the blood, okay, from the blood, that means when water level comes down, okay, loss of fluids from the blood. All of you are here with me? Yes. What it is going to do whenever such a situation happens, there is going to be stimulation. Okay, what it will stimulate? It will stimulate the hypothalamus. Body will stimulate the hypothalamus, telling there is loss of water in the body. You please release something that will stop this. Okay, that will stop this. Otherwise, the person will go through dehydration. Okay, so it will stimulate the hypothalamus. Hypothalamus indeed will stimulate your ADH. Okay, ADH or your vasopressin. Okay, release of vasopressin will be stimulated. Now what is vasopressin going to do? We have to make sure that the loss of water is less. Okay, that means diuresis condition, students. This is called as diuresis condition. This happens in people who have diabetes mellitus, okay, who have more glucose level in their body. They will lose a lot of water. So first thing vasopressin is going to do is going to do exogenous intake of water. That means going to activate. Okay, it is going to increase that is going to, what they are going to do, students, they are going to exogenously activate your thirst mechanism. Okay, it will activate thirst mechanism. So when you activate thirst mechanism, what happens? You will feel like drinking water. Second thing, what it will do, it will help in reabsorption. Okay, helps in conditional reabsorption or normal reabsorption. It can ask DCT and collecting duct to do conditional reabsorption as well. So this way, what is going to happen? Water intake is going to happen. Okay, water intake will happen. Here also, water will be taken in. Okay, water will be taken in. Understood? Any doubts? Yes. ADHA mechanism is understood. Okay, so there's going to be a feedback mechanism that will stop the ADH, because too much water also is a problem. Okay, too much water also is going to be a problem. Understood? Then the last mechanism is ANF mechanism. Students, actually ANF mechanism works against RAS mechanism. Yes, you heard me right. It works, okay, it works antagonistic, okay, antagonistic to RAS mechanism. So it works against, or let's use the word opposite, or you can write the word antagonistic, okay, opposite to RAS mechanism. Why am I telling this? Because students, here the problem, okay, here the problem is going to be high BP. When you see your NEET question paper, okay, high BP, okay, high blood volume, high GFR rate, ulta of RAS. Okay, when everything is high, ANF is going to work. Clear or not? Okay, so ANF is actually full form is going to be atrial natriuretic factor. Atrial natriuretic factor, which is actually released by the heart. Released by whom? By the heart. Clear? नहीं आती है सायन, कितना बार बोलूंगी, नहीं आती है मेरे को हिंदी. You go, please find a Hindi class and go study there, dear. Why are you wasting your time here and scolding English people? Okay, so students, what is going to happen? This ANF main issue is going to be all of this. Okay, so it's going to work against it. We can say that they're going to do vasodilation. Okay, they're going to do vasodilation. Okay. So here, students, these people will do vasodilation. Okay, vasodilation of the efferent arterioles, and this way what is going to happen? The blood is going to go out faster. Okay, so high blood flow in the atria, the ANF will be released, which will do vasodilation. Automatically, when vasodilation is done, your BP will become less. Okay, BP will become less because blood will start, more space for the blood to flow. Now imagine if the blood is flowing. So students, here imagine blood is flowing at this is the radius. Okay, this is the radius at which the blood is flowing. Don't you think the pressure is going to be normal? Okay, oh, is it really? Okay, so students, if the blood is flowing like this, you can understand that the blood is not going through a lot of pressure. Okay, so this is vasodilation. Okay, but imagine the same blood is flowing through a smaller surface area. definitely the BP is going to be high, because the blood pressure, the pressure exerted on the walls of the blood is going to be high. So here what is going to happen? Vasodilation is going to happen. Okay, vasodilation will happen, automatically reducing the BP, reducing the GFR. Okay, clear or not? Okay, done. So this is vasoconstriction. So you can see how much surface area is there. It's pretty less. Okay, it's pretty less here, but here it is more. Clear? Oh, okay. Clear. ANF is clear. Atrial natriuretic factor. Perfect. Done? Yes or no? Okay, done. RAS mechanism done. Okay, diuresis problem. Okay, done. Loop of ending. Students, one more thing I'll teach you. Wait. Flow of blood. Okay, that's the last topic. Then we will go for urine formation. Okay, I'm just checking your NCERT as well. So students, how does the blood flow? Okay, flow of, I'll erase this, sir. Oh, all dogs got activated with energy. Flow of blood. Okay, flow of blood. How does the blood flow? Anuja is a regular student here. We'll never block her. Okay, so look here. Students, aorta. Okay, from aorta, the blood is going to enter into the renal artery. Okay, renal artery. Okay, sure, Rathis. I will definitely do it, so that they can put their pellets and, you know, on my white color top. Afferent arterioles. Okay, afferent arterioles. Now students, from afferent arterioles it's going to go into the efferent arterioles, but before that what you have to remember is, you have to remember that it is going to go to the glomerulus, right? From glomerulus it is going to go to the peritubular capillaries. Okay, peritubular capillaries, right? So it is going to go into the efferent arterioles. Okay, from efferent arterioles it is going to the peritubular capillaries. Then students, from peritubular capillaries it is going to the renal vein. Now how many of you can relate this to? How many of you can relate this to? Tell me. Aorta, renal artery, efferent arterioles, glomerulus, efferent arterioles. Now students, how many of you can relate to the fact that from peritubular capillaries? Wow, notes is nice, no? Today it's very colorful. Okay, wait. Look here. This one, blood is entering from the renal artery. Okay, from the aorta, main aorta, it is entering the renal artery. From renal artery it is entering into the efferent, efferent to glomerulus, glomerulus to all the way up to the peritubular capillaries, peritubular capillaries to renal vein. Can you understand here? I told you this is the connection, so this question will come. That's why this connection I have made. Understood? That's the flow of blood. That's the flow of blood. So the renal vein is going to have deoxygenated blood, but it's going to have the non-toxic blood. Okay, after purification, non-toxic blood. Done. Okay, done. Yes or no? Yes. So that's it for today, students. That's our end of today's class. Okay, so we have completed half of excretion. Now we have urine formation and small portions, micturition and other things, so that we will do in tomorrow's class. Okay, so see you all in next class. Thank you so much for joining. Okay, and main notes I have already sent it on my telegram from Vedantu NEET English when I write it on the board. So these are all these notes cannot be saved as well, okay, because it's written on the PDF. Got it? So thank you so much for joining. See you all in the next class. Tada, bye bye.